improvement of single model methods. They also leveraged new developments in deep learning, so more complex representations of structure using graph neural networks, convolutional neural networks, etc. But the most exciting development in my eyes is this kind of fully integrated self-assessment with the example of AlphaFold, where really kind of you have this multitask deep learning model, and quality estimates is really just one of the outputs beside the coordinates. So in principle, it has full access to all the information that is used for modeling to predict these quality metrics on a per residue level. Now, if you read the paper, it sounds a bit like, yeah, it was just a pure convenience to train just everything at the same time, but it really kind of closes the loop, because quality metrics should be an integral part of modeling itself. So this is really kind of the natural evolution of QE that's basically all together. So for per residue assessment is still consensus is going strong. It was absolutely top. So the baseline predictor from Andrei was pretty good. Now off to new frontiers, assemblies. But first, we want to look what this now means, these new developments in per residue predictions that I claim are now so super accurate. What you see here, I couldn't resist, still consensus. It's just basically a re-implementation based on LDDT. So it's all versus, also basically what you're doing is you compute LDDT of a model with respect to all other models and just take the average on a per residue level. And just for speed purposes, this predicted LDDT is only based on C alphas, but it closely correlates with actual LDDT. You have here this characteristic line that comes from a stereochemistry checks that LDDT applies and just punishes for residues with stereochemical irregularities. Now, yeah, it's fantastic. I mean, C8, that's good. So just remember. Becomes relevant for the next slides. Now, there is no real ranking you can make if you look at the top methods. So what you see here is really just all based on LDDT, have a very basic evaluation based on Pearson correlation, absolute deviation, etc. that are just stacked on top of each other. So just as a simple proxy for how good these methods perform. So it turns out that it's really hard to distinguish anything. And it becomes even more, well, clear if you look at the small plot here on the right. So what you see here is kind of the sum of scores, which reflects these bars. And that's kind of a proxy for overall model quality, the average LDDT on a per residue basis. And what you see is it's also to a certain degree is a dependency on the underlying data. So it for sure is beneficial for Pearson correlation if we have some crap in there, right? So basically, if you're lower down here, you have more opportunity to go to the right. So it's really just this per method data dependency. So it behaves a little bit like a Pareto frontier. So there's no clear winner, and that's also kind of the message. So they're all pretty good. If you then directly compare it with what we saw before, consensus that I claimed is pretty good. It's there. It really is. So these estimates on a per residue level are really kind of comparable to what we get from a consensus method. So that's kind of the first conclusion. Let's go to assemblies. It becomes a bit more noisy. So here the exact same plot if you look at the self-assessments from assemblies. But what is interesting to see is also if you kind of separate a bit the residues into classes. So what you see here is core surface interface. So meaning that core residues tend to have highest, so it's easiest to predict qualities for core residues. It becomes a bit more difficult for surface residues. However, we don't know how much this actually is a method problem or just flexibility from the structure itself. So you get some more noise in the actual target value. And then it goes further down for interface residues. And here interface residue is as seen in the target. So we just go for the target, specify what is the interface residue. So this bar goes down even further if you use for this classification the actual model So to specify what is the interface residues. So we see there is a problem, and that's why also for this CASP we said, okay, so let's look at assemblies. So there were 3 tasks. 2 of them is global. So one task was really kind of predict global score. That is fully superposition based. So meaning you have to get the domains in the right orientation, etc., which we call score. And then QSCORE is really kind of give us an accuracy measurement of that is only interface based. So the accuracy of the interfaces themselves. Here is the scores that are used for this QSCORE category. It's QSCORE and DocQ, at least DocQ you heard already several times of. QSCORE is a contact-based score that is weighted in a sense that contacts that are close in either the target or the model get a higher weight, and contacts that are further away in both are a bit decreased in weight. It's symmetric, meaning it doesn't matter which is your model and which one is the target. And down here is DocQ developed in the Wallner lab, and it kind of takes the measures that are common in Capri and combines them to a score that nicely reflect these success categories in Capri. Now, the problem you have in DocQ is it's on a per interface basis. So I also had correspondence with Björn Wallner, and we were like, yeah, there is no real ideal way, because I mean, you have maybe this tiny little interface that is super crucial for the overall assembly, but still, in the end, it ended up to be just a weighted DocQ, so weighted by the number of native contacts. So big interfaces contribute more than smaller interfaces. That's just trade-offs we have to do. If you now go for TM score versus QS score, you see it's terrible, which is to some extent expected because they look at different things. So you have these kind of things, just huge assemblies. Where you see H1114, it was already discussed in the assembly modeling session. So you have these 4, let's say, domains or subunits that are actually easy to model because there is a template for those. And the real challenge was to get the overall orientation right, meaning get this stuff in the middle right. But nevertheless, all those interfaces are correct. Now, if you run this through TM, so through US align as we used it, you cannot superpose that thing because the orientation of these guys is totally wrong. But now if you do a QSCORE based chain mapping, it still catches that actually most of these interfaces are correct. So you really need to look at both. So you need to look at the actual interfaces, but also at the overall orientation. So that's also why it's kind of useful to have both aspects. Then you have other outliers. So here in this case, you see a bit the difficulty of the chain mapping problem. So tools use greedy approaches, so not like optimal approaches. So this is a perfect model. It's absolutely beautiful, but in this case, US Align messed up the chain mapping. So this would get a GDT of, I don't know, 0.99 or something. So just very, very high. But the TM score is low because one chain mapping was actually swapped. But that's just what happens. We're not living in a perfect world. Now, global target value. So there's 2, oligo GDTTS and TM score. Oligo GDTTS is not GDTTS as computed with LGA or something as usual, but rather we do a chain mapping and then use a simple CUPSH superposition on the aligned C alphas. So the problem there obviously is if you have outliers, you may expect superpositions that are pretty bad. It has shown to be surprisingly robust towards that, and then you just compute GDT with the common thresholds. TM score should be very well known. What I want to point out is this D0 parameter, because D0 is really a parameter that the idea is that for big structures, you give it a bit more slack. So meaning you allow a bit larger deviations for large structures. So this is why it's length dependent. But other than that, you can interpret it as if you do a superposition and all the alpha positions are roughly within that distance, you get a TM score of 0.5. So that's just a rough interpretation of it. You can plot them against each other. First thing you see is you have this this trend. So it's harder to get a high oligo GDTTS than a TM score. And then again, you have outliers. So one source of outliers is highly target specific. And when we look at it, for example, large complexes, when you run it through US-Align, you have these zeros of more than 20 angstrom. So you give it a lot of slack, and that's way beyond the normal thresholds you have in GDTTS. So this is why you have this shift here to the right that is common for the big guys like H1114, H1111, and there's a couple more that tend to be a bit more on the other side. Then you again have, so there seems some targets that are prone to chain mapping issues, because you wouldn't really expect the GDTTS way above this line here. So that's then again a chain mapping issue. So enough talked about this chain mapping. So what is behind it? So there is QSCORE already around. It has been used in previous CASPs. Was a bit depressed when I saw Andre's results that suddenly many of them just failed because of complexity. So the alternatives were using US-Align or QS-Align, and there are other methods, but most of them seem to be based on rigid superpositions, and this would make this QSCORE evaluation impossible. So yeah, so something new had to come up. So this is just a problem statement. You have a target, you have your model, and in the end, you want this kind of optimal assignment that in this case is super trivial because you have a high degree of symmetry. But the problem really is if you have to exhaustively enumerate this thing, you have a complexity that is just terrible. It's factorial. So you have factorial complexity in assigning these model chains into the target. So that's pretty bad. Now, so you can leverage some features of LDT or QSCORE that they're pairwise decomposable. So I can just start with a random assignment, and then the expensive part, the actual score computation, what you can do is really you can compute it. You can do a lot of caching. That's what I want to say. So it's basically these two chains in the target are mapped to these two chains in the model, which is obviously terrible, but you still need to compute how does it look like. But you can do a lot of caching. Now these pairs, the number of these pairs that behaves polynomially, so it's not factorial. It's still terrible, but not as bad. So you can cheat your way a bit up until you hit the brick wall of factorial complexity for the overall problem. Now, we can cheat our way up to roughly 12 chains. So that's for 12 chains, that's runtimes around 1 minute on a laptop. And for the bigger things, we use heuristics similar to the ones used in US-Align. So you start with a seed, and then you start to kind of expand your chain mapping. In this case, you just can afford a bit more sampling in the hope that it becomes a bit more accurate. So yes, there is an efficient solution. QSCORE evaluation would be impossible without. There is new developments in LDTs. So on the way, it was then also applied on RNA, on ligands, etc. So you saw it during the conference. Now the actual assessment. So thanks for all those who really looked at these targets, because so then I really could build on that work and just use them. And in the end, what we ended up with is roughly 10,500 models for which we had predictions from 40 targets. Wrong stoichiometry. I wanted to kind of kick out the bias when suddenly you have models with less chains. In one target, it was actually advertised as an 10-mer, but actually only 9 chains were resolved. So basically that full target was kicked out too. So we end up with 40. And we had quite a few groups. So what you see here is really the number of kind of predictions that we have for the score and QSCORE category. And basically the number of targets where we have at least 80% of the scores. So the idea really was, it was this new category, methods were experimental. So I don't want it to be too stringent and to punish too much for missing predictions. So this is really kind of the inclusion threshold for participation. There is this AC, which is, again, can resist, just assembly consensus baseline. In case of SCORE, it just returns average GDT to all models in the ensemble. In case of QSCORE. Whoa, that was one too much. So that's a ranking. There's a lot going on in this slide. So it's really kind of what we do is we compute metrics like Pearson correlation, Spearman, ROC AOC, loss, and so on a per target basis. We for one group, we average all the values from each target into one value, and in the very end, we compute the set scores. And that's what we then sum up. There is some things like only consider targets with at least one model that is decent, and negative set scores are set to 0. And I didn't want to go down the road of what is now a class threshold for AOC, so I just defined it as the top quartile is positive. So this is just the details of the analysis, and this is done for both scores. So you see AC is shockingly high. What I have to say here is that it's awesome for correlation measures and ROC AOC, but if you look at loss, it's not so good anymore. So we can kind of turn the thing around, and suddenly we have different methods on top, and AC goes quite a bit down. So let's say for 2/3 of the targets, method Van Clavas is able to pick something that is useful. Similar for QSCORE, here AC is actually on top. So these plots are basically on the web, so I don't need to go over them too much. But again, other methods catch up when it comes to the loss measure. Local, so we told predictors, okay, here we have the model. These are the interface residues, or more specifically, we told them how to find those interface residues, and they then had to predict scores of how accurate they believe this interface residue is. Has been evaluated with LDT and CUT, so there is no explanation needed. They behave neatly. Then additionally, there is these PEP scores. Before going into it, I just want to talk about some features of this plot here. When you can compare LDT with any of those weird scores, there is a decent correlation. But you have this nasty distribution here to the right. And this comes from the fact that LDT does not punish for added contacts. Meaning if you have in the model a wrong interface, that does not get punished in LDT because it's not a symmetric score. So we needed something else to counteract this, because especially in the case of interfaces, that's quite important. So here is the logical expression, but in the end, what it does, if you're interested in this residue R, what this logical expression is, it defines local patches on the same chains and the other chains. And then on these patches, you compute DocQ and QSCORE. Again, similar amount of models. Some more models have been kicked out because some models just had no interface at all. And there were, again, quite a few methods that participated. So 13 groups were evaluated. There is no baseline. And there is one clear winner. It's GUI JunLab RocketX. So there is these 4 scores, and let's say ZLDTs, and really a combination of Pearson R, Spearman R, and ROC AOC. So just the correlations are a bit weighted down because they're basically the same. So I slowly come to the conclusion: the target matters. If you look at average values, let's say correlation measures or whatsoever, it really it's not spectacular. We would expect more from tertiary structure. So what you see here is really a random selection of methods that performed well in QSCORE. And it's really just the Pearson correlation between this QSCORE prediction and QSCORE. And then you see here this whole group of targets where everything in the world is in order and everything is fantastic. I mean, that's spectacular correlations. And then for many targets, it just drops. So there seems quite a target specificity. What we saw is nanobodies are difficult. We saw that before in talks. And indeed, 4 out of these 5 nanobodies are here in this area where we have just no clue what's going on. If you look at this guy here, that is good. So someone told me there was some template. So it was actually also for the modeling side a relatively easy target. And what you see here is really just the reference here a bit fat and the nanobody in red. And I just took the nanobody as a reference for superposition. So you have this nice cluster of models that really clusters around the nice solution. So consensus is just super happy and you have, you know, your correlation of close to 1. But interestingly, also, if you take a completely single model method that does not know anything of the ensemble, the good good confirmations really cluster here on top. So that's really everything is, the world is in order. So that's pretty cool. So if you now look at this case here, just as another example, things become a bit more messy. So many people predicted something in the cloud over here. And then obviously the consensus method jumps on it because that's the biggest cluster. But the single model method here, okay, I agree. RocketX had a bit of luck. We also have to say that, but it picked out one of the top models. And also like Woroyev and other single model methods was able to pick a good model here. But everything that looks a tiny bit at consensus was totally lost in this case. There is another interesting target. No, there is one more thing I want to say here. So there is 5 of those nanobodies. And there was basically one method that was really good in picking out decent models. And that was kind of this Venklovas method. And 4 out of 5 cases, they picked a decent model. The other, so the 2 single model methods, Woroyev and RocketX, and I think they picked 2 or 3, which is also quite good, but everything else then just succeeded in that easy case. So there is more work needed on both sides, modeling and QE. So then there is the last example, there is a bit more food for thought. So there is this guy here, where you have in red, that's the reference structure, and then you have a big bunch of models that has a completely different conformation. The domains are perfect and everything, but there's a massive rearrangement. And the paper that came out in October described the structure as an inactive closed state. Now, I don't want to speculate too much, but what in the paper they propose as a model of function to bind DNA is pretty much such opening up that we see here. So the question is it really just a problem with the underlying structure? So, and with this, I really want to conclude, and also with the message that in quaternary structures, we may observe even more issues with flexibility, be it in terms of structural flexibility, but also in terms of stoichiometry, etc., than what we saw in tertiary structures. So that's kind of the big trouble we're facing there. And with this, I want to really conclude. So the Schwede people, so there is Gerardo, who did the self-assessment number crunching. So thanks. Then obviously Thorsten for lots of input. Then everyone who was involved in really looking at these oligo targets, and then obviously Andri. And the whole code to do this assessment is available on our local Git instance. So you can basically download it and reproduce everything. You cannot download some of the targets though. So you can ask me for the raw data tables to start Jupyter notebooks to kind of reproduce these plots. And yeah, that's it. So thanks to you.
Thank you very much, Gabriel. Very crisp, and thanks for staying on time. We have time for questions.
Hi, yeah, it's just a question about the chain mapping code.
Yes.
So are you going to make that available? Because we also had a problem with chain mapping.
Yes.
We came up with a solution based on the sequence matching. Because we had the target sequence, but are you going to make that available?
Mm-hmm. So in principle, the code is online and can be pulled. But still it's considered develop. It has to go to, you know, release cycle, etc. But we are really pushing out because you're not the only one who needs this code. In principle, you can go to this page here And there is a singularity container solution described how to set up to access the whole scoring that was used for the EMA, including chain mapping. So you get the information there. But we need to push it out. Yes, there was just too many different things that were.
Hi, Gabriel. In your self-assessment in the beginning, you confused me a bit. So what you're saying is it's much easier to predict core residues than surface residues, but it's most difficult to predict interface residues. And what I didn't get is it a prediction problem, or is it the underlying data is messed up because there's too much flexibility in there? Because I would naively expect that interfaces are between core and surface, but not worse than everything else.
That is totally true. And I totally also have the feeling that it's the underlying data that is messed up. I mean, yeah, so I mean, you may have just wrong interfaces, and maybe the target value, I mean, we saw LDT is for sure not the optimal target value for interfaces, simply because it does not punish for added contacts, and all these things start to add up. So it's not only a prediction issue, but also the underlying target value where we need to think of. So that's a problem with several aspects, just, yeah.
Just a comment about it, because you also looked at interfaces coming from cryo-EM, right?
Mm-hmm.
And what Tom showed yesterday is that there are some errors sometimes in the target as well.
Wow.
So you're comparing to, and I think this is why Capri actually didn't look at some of the targets, because they were at lower resolutions, or there were problems with interfaces.
So I mean, you're talking now full sequence shifts or something?
No, but position of, it could be position of sidechains that change the contact.
There we go.
Orientation of sidechains and so on.
So a lot of sources for.
I mean, it's worth doing this analysis maybe without the cryo-EM targets, just to see if you get the same thing.
Okay, if no further questions, let's thank again to Gabriel.
Yeah, thanks.
And now we are moving to our first presentation, should be online presentation from Guijun Lab Rocket, and Guijun Zhang will present it.